Assalamu alaikum. Dear students, I'm here, Muhammad Tajammul Khan, lecturer in botany, University of Education, Lahore. Today, we will discuss a topic that relates to our subject microbiology. The course code of this subject is BOTN 4128. In this subject, today we'll discuss a topic that is the brief history of microscopy and units of measurement. So here we have a content that we divided it into two parts. In first part we discuss about the brief history of microscopy, and in the second part we discuss about the units of measurement. So here we have the brief history of microscopy. So let us view an invisible world. The objects around us that are too small to be seen with the naked eye. So the mentioned timeline in this slide provides a look at some of the key advances in the history of microscopy. So first of all, we started with 710 BC, in which that is we referred as a Namrud lens. So a piece of rock crystal someone discovered that looking through a crystal made things look larger. So initially crystal was called a magnifying glass, and later on, due to its similar shape with the lentil seed, later on it was called as lens. So in 710 BC, a piece of rock crystal may have been used as a magnifying glass or as a burning glass to start fires by concentrating sunlight. After this, in 1000 AD, the first vision aid called a reading stone is invented. It is a glass, sphere in shape, placed on a top of text, which it magnifies to aid readability. So in 121 AD, a book written by a Muslim scholar named as Ibn al-Hassam, it mentioned some related to optics. Eventually, it transforms how light and vision are understood. And he called his book a book of optics. Later, in the 13th century, a inventor, an Italian inventor, made the first eyeglass, allowing wearer to have a magnification. The name of scientist is Salvino di Ermate. So eyeglasses were also called spectacles. So in 1590 AD, a scientist named as Scaurus Janssen and his son Hans Janssen produced a first microscope by using multiple lenses tube. They observed that viewed object in front of a tube appeared greatly enlarged. In later in this lecture, we discuss in detail regarding the earlier microscope. So after that, in 1609, a scientist named as Galileo developed a compound microscope with the convex and concave lens. So the previous microscope are very hard to use. So here these are the figures that showed the previous microscopes. So these are very rare surviving simple microscopes. So after the invention of glass, the Romans started experiment with it and testing it. And they found that if the glass had a thick middle but was thin at the edges, object would look larger when this lens was held over them. So these early forms of lens were referred to as magnifiers. So in 1590s, a Dutch spectacle, Zacharias and his father Hans Janssen. Actually, these both are eyeglass makers, and they are the inventor of a first compound microscope. So he produced a compound microscope with simply a tube with lenses at each end, and the magnification of these early scopes range from 3x to 9x depending upon the size of diaphragm openings. But their compound microscope was not so good due to poor lens quality. So the early compound microscope could only magnify an object 20 to 30 times than its normal size. But he observed that if one put a small object in a tube containing several lenses, the object would appear very large. when at the end of tube and was much more enlarged than when a simple magnifying glass was used. So here we have the images. The first microscope, that is by Jensen, was simply a tube and with lenses at each end, one from this side and one from this side. So the magnification of these scope ranges from 3x to 9x depending upon the size of diaphragm. So in next they pair together these lens for magnification, or the function of simple early Italian compound microscope. So the function, or these form the earliest compound microscopes, and its main function of this microscope only to magnify the object. So now in from 1635 to 1703, a scientist named as Robert Hooke, uh, the first big microscopy advances came in this time when Robert Hooke published a Micrographia, a collection of copper plate illustrations of objects that he observed with his own compound microscope. Actually, Robert Hooke is an English physicist, and he looked at a silver of cork through a microscope lens and noticed some pores in it. So he was the first person to use the word cell to identify microscopic structure when he was describing cork in his Micrographia. Here you showed his Micrographia here. This is the Micrographia of Robert Hooke, in which he used the word cell to describe the features of plant tissue. Actually, he used a cork from the bark of an oak tree, and he observed here these are the observation of Robert Hooke, in which he observed a small cells in the cork cell of oak tree, and that is discovered by Robert Hooke through his compound microscope. And he observed the cell at the magnification of 3x. Sorry, it's 30x, not 3x. So after that, he improved his microscope and he changed and took some changes in his compound microscope. So this modified microscope actually contained a monolocular eyepiece. So he used a tube that is made up of wood, and in addition to this he used a stage of holding a specimen and a glass globe that is full of water to concentrate a light on specimen. And in addition to this, for a light source he used an oil lamp. So after this modification, he improved his compound microscope. Simply he used a wooden made tube that contains lens on each side, then a stand on which this apparatus is the stand. Then he used a monocular eyepiece, a stage for holding specimen. Then he used glass globe for water to concentrate light, and as a light source he used oil lamp. So by the observation from this modified compound microscope, he observed these Hooke cells, these cork cells from a cork of oak tree. As you observe these cork cells here, the first time he used the term cell for these small units that he observed from cork cells. Then he observed different insects from this microscope. Here we have a micrograph of these insects that he observed through his modified or improved microscope. Here he observed the cork cells. These are the cork cells. So now the next era is belongs to Anton van Louis van Hooke, started from 1632 to 1723. In the same century, Englishman Robert Hooke was acknowledged as the discovered the smallest basic unit of an organism, the cell, as we discussed earlier. Anton van Louis van Hooke, he was also recognized as the first person to use a microscope with 3 lenses. So the configurations used in today's microscope. So Louis van Hooke was the first to see and describe bacteria in 1674. He also observed yeast from plant and the living things in a drop of water and the circulation of blood corpuscles in capillaries. Louis van Hooke made simple microscope, and he was not the first person to build a microscope, but the microscope he did build were the best one of that time period. So Louis van Hooke was the first person to describe bacteria that he obtained from teeth scrapings, protozoans, and from pond water, and he also helped to prove the theory of blood circulation. Louis van Hooke refined lens grinding so that the living things could be seen through the microscope. Actually, he was so much inspired from Hooke's micrographia, and he gained so much inspiration from reading of Hooke's micrographia. So the Louis van Hooke's microscope was really a powerful magnifying glass rather than a compound microscope, because Louis van Hooke used hand ground lenses that could magnify an object up to 200 times. So on the behalf of this, he observed animal and plant tissues, even blood cells, minerals and fossils, and much more. He used his microscope to discover nematodes and rotifers, microscopic animals, and these instruments were unlike what we commonly think of microscopes today. So consisting of a single tiny lens, and there were actually very powerful magnifying glasses, the figure I showed later in this presentation. So here we have the figures that he observed here from his microscope, a magnifying microscope. Here, first of all, here we have the picture of that microscope that he used to observe different organisms, specifically bacteria or blood cells. Here he observed some protozoans, spermatozoas, and protozoans that he observed through this microscope. So the major thing that he gained in his microscope, he refined lens grinding so that the living thing could be seen through the microscope in a very clear and a very magnifying manner. So it was difficult to focus one of Louis van Hooke's microscope instead of changing specimen. So he built a new microscope for each specimen, leaving the previous specimens and microscope together. So when the foreign investigator came to Louis van Hooke laboratory to look through his microscope, he made them keep their hands behind their backs to prevent them from touching the focusing apparatus. So after that, Louis van Hooke described his first observation in a letter that he sent to the Royal Society of London in 1676, in which he mentioned that he observed bacteria and protozoa in water. He kept his technique secret. However, even today we are not sure of his method of illumination. So although he used indirect lighting with light bouncing off the sides of specimen rather than passing through them, Louis van Hooke was also unwilling to part with any of the 419 microscopes he made. It was only near the time of his death that his daughter, at his direction, sent 100 of them to the Royal Society. Now it's time for Industrial Revolution. At this time, not much change in the basic microscope design occurred, but better lenses were crafted. Using pure glass and using different shapes to solve the problems like color distortion and pure image resolution. So in this era, a standardized parts would be available and that were interchangeable with other microscopes that lead to the mass production of this item. And it's in this era there is a trigger to drop in price of microscope so that the access of this item or access of microscope will be easy for scientists or for botanist. So in this era, a new discoveries related to a clear image will be would be possible, and approx in 1880 a modern microscope were being used. So with the advancement of technology and improved optics, the compound light microscope came into being, and this is the most modified form in the Industrial Revolution. In addition to this, In this era, in late 80s, a scientist named as Ernest Abbe discovered the oil immersion lenses prevented light distortion at highest magnification powers. So these are still used today on 100x objective microscopes. Now this era had to end, and there is the beginning of 19th century. And in this century, a pair of German scientists invented a most advanced form of microscope, that is electron microscope. So in this kind of microscope, he directly use a beam of speeded up electron at a cell sample. So as the electrons are absorbed or scattered by different parts of cell, they form an image, and that can be captured by an sensitive photoplate. So this model enables scientists to view extremely small parts magnified as much as 1 million times. Now here we have the second part of this lecture, that is units of measurement. In our daily life we use different units of measurement, depends upon the physically or practically usage of the item or subject. So the most common units that we used in our daily life, these are centimeters and the millimeters. Then we have micrometers, nanometers, angstrom. So we have a smallest measurement unit, that is angstrom, and majorly we used this unit in microscopy. And here we have the values of these units. Here the centimeter is the negative power of 10, this one. Similarly millimeter is the negative 3 power of 10, negative 6 power of 10 is micrometer, the negative 9 power of 10 is equal to nanometer, and then negative 10 power of 10 is equal to angstrom. So these are the various units that we used for calibration or for measurements in our daily routine matters or daily routine life. So basically these units are used to classify and describe very small objects. So one of the most common microscopic measuring unit is the micrometer, and that we also called as micron, which is the 1/1,000,000th of a meter. So here we have a unit that is majorly used for measurement in microscope or in microscopy, that is micrometer, or it is also called as micron. So here we have a scale 1 millimeters. Here we have a scale that is 1 millimeter. Here this is a 1 millimeter part. 1 millimeter, that is equal to 1000 microns. So it's mean 1 millimeter is equal to 1000 microns, and from this to this we have 1000 microns. And how big is micron? Here you observe the micron, how big we have a micron. So human hair is almost about 17 microns in diameter. So here we have a 1 micron, then we have a 15 micron, then we have a 13 micron, and human hair that is also most about near about 18 to 17 microns. So it is the too much smallest unit that is used for microscopy. So here we have two terms. One is magnification, the other one is resolution. So what is magnification? First of all, discuss about magnification. Magnification basically is how much bigger a sample appears to be under a microscope than its real size. So before this, by using more lenses, a microscope can achieve greater magnification, but this does not mean that more detail can be seen. The detail of any specimen would be visible depends on the resolving power of the microscope. So it's mean that the smallest separation at which two objects can be distinguished. So the resolving power of microscope is ultimately limited by wavelength of light. Later on we discuss about the wavelength of light. So to improve the resolving power, a shorter wavelength of light is needed, and sometimes microscope have blue filter for this purpose, because the blue light has shorter wavelength. So the magnification, how much we can enlarge the image, and when we obtain the overall magnification, we simply multiply the objective lens to eyepiece lens. So the magnification of objective lens, when multiplied with the eyepiece lens, we obtain overall magnification. The next thing is resolution. What is resolution? Resolution is the simply we can say that it is the ability to distinguish between 2 points on an image. So resolution is limited by the wavelength of radiation used to view the sample. So this means that when the object in the specimen are much smaller than the wavelength of the radiation being used, they do not interrupt the waves and so are not detected. So the wavelength of light is much longer than the wavelength of electron, so the resolution of light microscope is a lot lower. So actually resolution reveal the sharpness we can get from the image. So now we discuss about the principles of microscopy on the behalf of which we operate a microscope. So first of all, we discuss about metric units. We already later, but I'll give you a briefing regarding metric units. After that, we observe the properties of light, and in the end we observe the properties of light with reference to the wavelength and resolution. So here first of all we have the microscopic units. Simply, microscopy is a technology for making a very small thing visible to the human eye, because the microorganisms are very tiny or very small, and the units used to measure them are likely to be unfamiliar to the beginning students. So we deal with a microscopic world as given here in this table. We already discussed that we have a unit, meter, then we have a centimeter. We have their measurements, then we have a millimeter, micrometer, nanometer, and angstrom. Angstrom is the smallest one, that is the 100 negative 10 power of 10. So first of all, we discuss one by one these units. First of all, we discuss about micrometer, that is commonly used in microscope, formerly called micron, and that is equal to 0.000001 meter. A micrometer can also be expressed as 10 to power negative 6 meter. Similarly, the second unit is nanometer, that is expressed as 10 to power negative 9 meter. And similarly, we have the third unit, that is angstrom, that and is found in much of the current and older literature, but no longer has any official recognition, and it is expressed as 10 to power negative 10 meter. So here in this figure, we observe the relative size of objects, and these sizes are shown relative to the metric scale. Named in red are organisms that study in microbiology, and in these, bacteria are much smaller in size than other bacteria. So the range of effective use of various instruments is also depicted from this figure. So here we have atoms that fall under here near about 0.1 nanometer. Then we have amino acids. He fall here near about 1 nanometer. So here we have different range. Here we have a DNA structure, proteins, ribosomes. Here we have a bacteria. Here, so here nearly about fall in the category from 1 micrometer to 100 micrometer. Here we have a viruses that have a very smallest size near about 100 nanometer. Then we have blood cells that near about 10 micrometer. Then we have larger organism, and in the end we have a dog which is fall near about 1 meter to 10 meter here. So we have a number of range in which different organism or different objects fall. So from this figure we get that from 0.1 nanometer to 1 millimeter, we easily observe the object through transmission electron microscope. Here transmission electroscope 10. Then from near about 10 nanometer to 100 millimeter, we observe through a specific microscope, that is scanning electron microscope. And over daily uses microscope, that is compound light microscope, that is observe the organism from near about 1 micrometer to 10 millimeter almost. And after that there is no need of microscope that need from 1 millimeter to 10 meter. So here we have the properties of light and object with reference to refraction and refractive index. First of all, discuss about the refraction. To understand how light microscope operates, one must know something about the way in which the lenses bend and focus light, or diverge the light, or converge the light. When a ray of light passes from a medium to another, refraction occurs. That is, the ray is bent at the interface. Later in the diagram we revealed it. After that, there is a refractive index. Refractive index is the measure of how greatly a substance slows the velocity of light. The direction and the magnitude of bending is determined by the refractive indexes of the two media forming the interface. When the light passes from air into glass, a medium with greater refractive index, it is slowed and bent toward the normal. A line perpendicular to all the surfaces as shown in next diagram. Here this is a glass and this is a air. So we have two medium, one is air, the other one is glass. So when here we have a normal, this is the perpendicular angle with reference to uh, air and glass. So when the beam come towards this or towards glass, as the light leaves glass, it returns to the air. From here we observed this is the beam of light that come towards the glass. This is the normal, and at this, so the beam move away from the normal. Why? Because the air, a medium with a lower refractive index, it accelerates and is bent away from the normal. The prism bends light because glass has a different refractive index from air, and due to this, the light strikes its surface at an angle. So as you know, light is made up of electromagnetic radiations or waves, and with properties such as wavelength and frequency, light usually behave like a wave and can be diffracted. So the wavelength here, the distance between two adjacent crusts. The one crust is here, the other one is here, and here, and between these two crusts, it is a wavelength. So the distance between this distance between two adjacent crust or adjacent trough of any wave is defined as wavelength, designated by a Greek letter lambda. So here light is electromagnetic waves, or so it possesses a characteristic or attribute of wavelength that that assist to diffract it. So these attributes of the light play a very vital role in the observation or in the observation of very small organism through microscope. So microscopy is basically depend upon the characteristics of light, then depend upon the object that we observed through microscope, and then depend upon the lenses. So this is all about for this lecture. So here we have the references that we get from different sources to make this lecture appropriate for our students. This is all about for this lecture. Thank you so much.